Recently, Hantavirus has become one of the most talked about health topics around the world, and I've done detailed research to bring you a full summary in this video. In this video, we'll take a closer look at how Hantavirus emerged, how it spreads, how it affects the human body, and why it can be so dangerous. We'll also talk about recent cases, scientific findings, and some of the lesser-known facts about this virus. Before we begin, don't forget to subscribe and leave a like to support the channel if you enjoy these kinds of detailed research videos. Now, let's dive into the hidden world of Hantavirus together. Genetics and structure. Hantaviruses have a complex genome made up of 3 RNA segments known as L, M, and S. The L segment produces the RNA polymerase enzyme that allows the virus to replicate, while the M segment encodes the GN and GC glycoproteins that help the virus attach to host cells. The S segment produces the N protein, which protects the viral RNA, and in some strains, it also contains the NS protein that can suppress the immune system. Hantavirus particles are usually spherical in shape, but they can also appear oval or tubular. Their size ranges from approximately 70 to 350 nanometers. The surface proteins embedded in the outer lipid envelope allow the virus to bind to cells, while inside the virus, there are helical nucleocapsid structures that protect the genome. One of the biggest mysteries scientists still have not fully solved is how hantaviruses are able to form such organized spherical structures even though they do not produce matrix proteins. Life cycle. Hantaviruses primarily infect endothelial cells that line the inside of blood vessels, as well as immune system cells known as macrophages. In addition, kidney cells, dendritic cells, and certain types of lymphocytes can also be affected by the virus. To attach to host cells, the virus uses surface glycoproteins called GN and GC. These proteins bind mainly to beta-1 and beta-3 integrin receptors, initiating the process of viral entry into the cell. After attachment, the virus enters the cell through different endocytosis mechanisms. Old-world hantaviruses mainly use clathrin-dependent pathways, while new-world hantaviruses prefer clathrin-independent pathways. Once inside the cell, the virus is transported into cellular structures known as endosomes and lysosomes. As the environment becomes acidic, the viral envelope fuses with the cellular membrane, releasing the viral genetic material into the cytoplasm. The RNA-dependent RNA polymerase enzyme then begins copying the viral RNA and producing new viral proteins. The newly produced genome copies are wrapped by N proteins to form ribonucleoprotein complexes. Meanwhile, the virus's surface glycoproteins are processed in the endoplasmic reticulum and split into GN and GC proteins. The assembly and release process of the virus differs depending on the hantavirus type. Old-world hantaviruses acquire their envelope in the Golgi apparatus and leave the cell through vesicles. In contrast, New-world hantaviruses bud directly from the cell membrane and obtain their envelope from the cell surface itself. Evolution. The evolution of hantaviruses mainly occurs through genetic mutations. These mutations can appear as single nucleotide changes, genetic insertions, or deletions.
These mutations can appear as single nucleotide changes,
are
genetic
usually
insertions,
associated
or
with a
deletions.
specific natural reservoir host.
However, this
and tend to
relationship is not
evolve
completely
alongside their
fixed for every
hosts over
hantavirus
time.
species.
However, this relationship is not completely fixed for every hantavirus species. Scientists believe the evolutionary history of hantaviruses is highly complex. This is largely due to frequent events such as host switching, cross-species transmission, and genome rearrangements. Because hantaviruses have a segmented RNA genome, genetic material from different viruses can combine together. This process is known as genetic recombination and reassortment. As a result, entirely new hantavirus variants can emerge. This genetic flexibility allows the viruses to adapt more easily to different animal species, and a wide range of ecosystems. Geography also plays a major role in hantavirus evolution. For example, studies have shown that Hantan and Sol viruses have developed distinct genetic lineages in different regions of the world. Transmission. Hantaviruses are primarily transmitted to humans through rodents. Mice and rats often carry the virus without showing any symptoms and spread it into the environment through their urine, droppings, saliva, and blood. Humans can become infected by inhaling contaminated particles in the air, consuming contaminated food, or through rodent bites and scratches. The virus is highly resistant to environmental conditions. It can survive for around 10 days at room temperature and even longer in cooler environments. This significantly increases the risk of transmission, especially in enclosed, unhygienic areas with high rodent activity. Farmers, forest workers, campers,
soldiers, miners, and people who work closely with rodents are considered high-risk groups. Contact with pet rats can also be a major source of infection. For example, in 2017, dozens of people in North America became infected with the Sol virus through contact with pet rats. Scientists believe that the Andes virus may be the only hantavirus species capable of limited human-to-human transmission. Close contact, saliva, cough droplets, and in some cases, transmission through breast milk or the placenta have all been considered possible routes. Additionally, some hantaviruses have reportedly been transmitted through blood and platelet transfusions. Research has shown that not only rodents, but also animals such as pigs, deer, rabbits, bats, and shrews may carry hantaviruses. This may help the virus evolve and adapt to different species more easily. Human activities also contribute to the spread of hantaviruses. Deforestation, intensive farming, poor waste management, and unplanned urbanization push rodents closer to human living spaces. Infection risks are especially high in war zones, refugee camps, and areas with poor hygiene conditions. Symptoms and clinical signs. Hemorrhagic fever with renal syndrome, also known as HFRS, is a serious disease caused by hantaviruses and usually progresses through 5 stages: fever, hypotension, oliguria, polyuria, and recovery. Symptoms typically appear around 12 to 16 days after exposure to the virus, although this period can range from just a few days to as long as 6 weeks. One of the most distinctive features of the disease is its effect on the kidneys. Swelling of the kidneys, excessive protein in the urine, and blood in the urine are common findings. Patients may also experience high fever, headaches, lower back pain, nausea, vomiting, diarrhea, and vision problems. After the initial symptoms, hemorrhagic signs may develop, including red or purple spots on the skin, internal bleeding, coughing up blood, and in severe cases, bleeding in the brain. Some patients may develop disseminated intravascular coagulation, or DIC, a dangerous condition involving widespread blood clotting that can become life-threatening. The severity of the disease varies depending on the virus strain. For example, the Hantan virus often causes severe illness, while the Puumala virus usually leads to milder infections. During the hypotensive phase, sudden drops in blood pressure and shock can occur due to vascular leakage. This is followed by the oliguria phase, where kidney failure develops and urine production drops significantly. Later, during the polyuria phase, kidney function gradually begins to recover, leading to excessive urine production. Full recovery may take several months. Some patients may develop long-term complications, such as chronic hypertension, kidney damage, and hormonal disorders.
Severe complications can also include respiratory failure, brain inflammation, pancreatitis, and inflammation of the heart lining. Infections caused by the Hantavirus and some Dubrava-Belgrade virus strains often result in a milder form of the disease known as nephropathia epidemica. Children generally experience milder symptoms, while infections during pregnancy may lead to more serious outcomes. Research also suggests that many hantavirus infections may go unnoticed because they resemble mild flu-like illnesses. In addition, repeated hantavirus infections have not been observed so far, which suggests that recovered patients may develop long-lasting immunity. Diagnosis. The initial diagnosis of hantavirus infection is usually based on the patient's symptoms and exposure history. Contact with rodents, staying in rural areas, or working in unhygienic environments are considered important warning signs. Common laboratory findings include low platelet counts, elevated white blood cell levels, impaired kidney function, as well as blood and protein in the urine. For a definitive diagnosis, it is necessary to detect viral RNA, viral proteins, or hantavirus-specific antibodies.
Kesin tanı içinse virüse ait RNA'nın, viral proteinlerin veya hantavirüse özgü antikorların tespit edilmesi gerekir. Enfeksiyonlar kendi konuları ortaya çıkabilir ve genellikle ömür boyu kalabilir.
Increase over time and can often remain for life. ELISA, immunofluorescence, and neutralization tests are commonly used in hantavirus diagnosis. ELISA is especially preferred because it is relatively inexpensive and can be used during different stages of the disease. Rapid IgM tests also provide practical and cost-effective diagnostic options. PCR testing is used to directly detect the genetic material of the virus. This method can help estimate disease severity during the early phase of infection and allows genome analysis to identify the specific hantavirus strain responsible for the illness. However, low levels of the virus in the blood may sometimes lead to false negative results. Prevention. The most effective way to prevent hantavirus infection is to reduce contact with rodents. Controlling rodent populations in homes, workplaces, and camping areas plays a major role in lowering the risk of infection. Keeping living spaces clean, storing food properly, and removing potential nesting areas can also help prevent the spread of the virus. People working in high-risk environments, such as farming, forestry, and pest control, are advised to use masks, gloves, and protective equipment, especially when cleaning rodent-infested areas. Ventilating contaminated spaces and avoiding airborne dust are also important safety measures. Hantaviruses can be destroyed by high temperatures, ultraviolet light, and certain disinfectants. Vaccines against some hantavirus strains have been approved in countries like South Korea and China, although regular booster doses may be required. Researchers are also continuing to develop newer and more advanced vaccine technologies.